All right, let's jump right into this explainer. Today, we're tackling Cambridge Biology 9700, specifically Section 6.2, the template strand versus the non-transcribed strand. You know, every single year, thousands of students walk out of this exam feeling totally confident about transcription. They memorize the textbook, they know their DNA from their RNA, and then boom, results day hits and they're shocked. The problem? They fundamentally misunderstood what the examiners actually want to see. We are going to fix that today. I'm going to walk you through exactly how to structure your understanding so you never drop these crucial marks again.
Just look at the difference between a flawed answer and a top-band response. It's honestly night and day, and it instantly reveals why normal revision just doesn't cut it. The flawed answer simply states that the strands are different in transcription. I mean, sure, that's true, but it's totally hollow. It tells the examiner absolutely nothing about your actual biological knowledge. Now, contrast that with the top-band answer. It gives this incredibly precise contrast of the template strand's physical role versus the non-transcribed strand's coding sequence. It's specific, it compares the two directly, and it proves you know exactly what the components do. That right there, that is literally the gap between a C and an A star.
So, taking a quick second, can you spot the exact grade-dropping error in that flawed answer? It completely misses the why and the how. Cambridge academic benchmarks don't just want you to state facts. They require logical, structured reasoning. You absolutely have to define the causal relationships. The assessment framework prioritizes analytical depth, way over superficial breadth. If your answers are just skimming the surface, you're leaving a ton of marks on the table, for sure. Let's rebuild this from the ground up. Okay, to really cement our understanding, we have to lock down the exact biological role of the template strand. It acts as the direct physical template for synthesizing a complementary RNA strand during transcription. The absolute keyword here is physical template. We aren't just talking about abstract code. During transcription, RNA polymerase binds to and physically reads this very specific strand. It's actively using it to lay down those complementary RNA nucleotides. You've got to explicitly state this physical action in your exam. It's the active machinery building that RNA molecule. And that brings us to the non-transcribed strand, which perfectly highlights the contrast. The non-transcribed strand is the DNA strand whose base sequence is completely identical to the synthesized RNA. Well, except for uracil swapping in for thymine, of course. But here is the massive distinction. It is not directly read by RNA polymerase. It just sits there. It holds the genetic code reference, but it's not the physical scaffolding being transcribed. Framing it as this identical genetic twin rather than the active template is precisely the level of detail the examiner is hunting for. Now, getting those higher order marks means you need to systematically identify the similarities and the differences between these strands. You can't just list how they're distinct. Both strands follow complementary base pairing rules, and they both share that classic DNA nucleotide structure, deoxyribose, the phosphate group, and a nitrogenous base. But they wildly differ in their roles regarding RNA polymerase binding and sequence matching. Pro tip, make sure you use comparative words like whereas or conversely in your exam to really lock down those comparison marks. In just a moment, I'm going to share a hidden examiner clue that completely flips how you should approach these questions. But first, you really need a procedural framework that makes that clue super obvious. Understanding the biology is literally only half the battle. The other half is deploying that knowledge under intense exam pressure. Without a structural plan, you'll just read a generic paragraph about RNA polymerase, and that's going to hardcap your grade. This is where the method mark strategy comes in. It's a strict 5-step sequence where you get marks just for showing your logical process. Step 1, deconstruct the question. Step 2, extract the relevant data. Seriously, do not ignore those graphs or text prompts. Step 3, select your analytical framework. Step 4, execute it with clear notation. And finally, Step 5, present your answer using that precise terminology we just talked about. This sequence is your ultimate safety net. It proves to the examiner you are reasoning, not just guessing. And when you hit the statistical evaluation phase, you absolutely must match your statistical test to the data type. If they ask you to compare the means, like looking at transcription rates across different temperatures, you're going with the t-test. If you're dealing with categorical data, maybe the presence or absence of a transcript, that's your chi-squared. And if you're tracking relationships between variables, like how temperature changes the transcription rate, you use correlation. Nailing this matchup is totally non-negotiable for those high-band practical marks. What's really fascinating here is the shift from just describing biology to the higher-order problem-solving that examiners really want to see. This is the deconstruct, solve, evaluate loop. You identify the given information, break it into logical steps, solve it systematically, check if it actually makes biological sense in the real world, and then make your justified recommendations. Making that jump from just defining a template strand to actually evaluating an experiment is exactly what separates average students from the elite.
So, how exactly do we apply this framework to evaluate actual real-world experimental evidence? Let's say an exam question throws a table at you, showing unexpected RNA sequences from a lab. If you just spit out textbook definitions, you score 0. You've got to use this framework as a lens to really interrogate the data.
Evaluating evidence quality means systematically stepping through a checklist. You have to identify the limitations, note the assumptions being made, find alternative interpretations, and assess the effect on the final conclusions. If there's a mutation in the template strand, don't just say transcription fails. Think deeper. What assumptions is the experiment making about how RNA polymerase binds? Could there be another reason the RNA sequence changed? How does all of this impact the final conclusion? This level of critical analysis is exactly what they are looking for.
Check out this warning straight from a principal examiner report. They say the classic mistake is providing generic answers not specific to the question context, and that you must always link every point to the given scenario. This is huge. If a question is about a specific plant virus hijacking the template strand and you write a generic essay on human DNA, you will lose the marks. You absolutely must anchor your theory to the exact scenario and organisms given in the prompt.
Honestly, we can boil the examiner reports down to a top 3 list of fatal errors. Number 1, using the wrong terminology for the wrong context, like calling the non-transcribed strand inactive, which is just biologically false. Number 2, forgetting to check your units and terminology. And number 3, which we just hammered home, providing answers that aren't specific to the context. It might actually help to memorize these 3 warnings and run through them before you put your pen down on a big 6-mark question.
Let's turn up the heat with a harder example and see how this plays out. A major error here would be just stating generic transcription facts, but the top tier, top band fix, that involves linking specific nucleotide sequence anomalies, say a missing uracil base, directly back to the provided experimental conditions, like a chemically altered template strand. That's how you actually synthesize your knowledge. It goes concept, then evidence, then scientific reasoning.
Here's a bit of an insider secret. To get the highest marks, you need a connected, conceptual understanding that you can transfer to unfamiliar, cross-topic contexts. Biology isn't just a bunch of isolated chapters. The non-transcribed strands code directly dictates the primary structure of a polypeptide from another topic. Examiners might link topic 6 to protein structure in topic 2, or even to cancer mutations. Drawing those conceptual lines across the whole syllabus shows a maturity that practically guarantees those top marks.
But here's a crazy thought. What if you perfectly understand the template strand? Your conceptual links are flawless, yet you still score a flat 0. How? Because you misunderstood the command word. If you beautifully explain the template strand, but the prompt actually asked you to evaluate it, 0 marks. Your brilliant knowledge gets blocked by a simple procedural mistake. The command word literally dictates the entire structure of the answer they expect.
So, the next time you're looking at a DNA sequence, ask yourself, are you just seeing a random string of letters, or are you looking at the actual physical machinery that dictates all of life? To make sure you decode those exact command words and translate your knowledge into exam success, be sure to subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE. Until next time, keep analyzing and always question the data.